Hello everyone. In this video, I am going to talk about processes of recombinant DNA technology. Let's recap about what is recombinant DNA technology. It is joining of genome of two different organisms, and hereinafter it is transferred to the host organisms to produce useful products for human uses, and that is called recombinant DNA technology. So there are various processes. So in this video we are going to discuss about what are the steps involved in this process. Where the DNA molecule is located? It is located in the nucleus of a cell. If we have to find a DNA molecule, first of all, we have to open up the cell. And the cell could be bacterial cell, plant cell, or animal cell, or fungal cell. If we are going to do genetic experiment with the bacterial cell, then culture the bacteria on the suitable medium and then harvest it. And the bacterial cells, they are treated with hypertonic solution. Bacterial cells are surrounded by a cell wall, you know that, and that is made up of peptidoglycan. And peptidoglycan can be digested by an enzyme, that is lysozyme. So we treat bacterial cell in the hypertonic solution containing lysozyme, and the bacterial cell wall will be digested, and the different macro and micro molecules which are present in the cells, they will be obtained. If it is a plant cell, to get the DNA from the plant cells, we can take any plant part of any plant tissue, and it is ground in the mortar, and the mixture is treated with the hypertonic solution. And plant cell is also having cell wall, and cell wall is digested by an enzyme, that is cellulase. So by treating this plant cell with the solution, that the plant cell wall gets digested and the biomolecules are obtained. Similarly, in animal cells, that the cell wall is not present and it is treated with hypertonic solution. In case of fungus, that the fungal cell is again treated with hypertonic solution and enzyme, that is chitinase. After treating the cells in this solution, we get the, for example, I am taking here bacterial cell. We get the different types of biomolecules, we get the different types of macro and micromolecules. These micro and macromolecules could be proteins, RNA, carbohydrates, lipid, and DNA. We get the following biomolecules, but the molecules of our interest is only DNA. So we need to remove all these molecules, and so these all molecules are treated with another enzymes. For the removal of protein, in the same solution we add proteases. For digestion of the dissolution of RNA, we add ribonuclease. And for removal of the other components, it is appropriately treated. All these molecules like proteins, RNA, carbohydrates, lipids, they are removed by appropriate treatment, and we get the desired molecule, that is DNA. After this, chilled ethanol is added to the DNA suspension. So here chilled ethanol is added. Since ethanol is dehydrating agent, it causes precipitation of DNA. When the DNA gets precipitated, using the glass rod, we remove the thread of DNA from the suspension, and this process is called spooling. So this is how we extract the DNA from the cell. And this was the first process. The second process, that is fragmentation of DNA by restriction enzyme. Suppose this is the DNA and we want this particular part of the DNA because here the gene of interest is present. So we don't need the entire molecule of DNA. We need particularly this fragment of DNA. So we need to remove the other undesired part, and for that enzyme is there, and that enzyme is called restriction enzyme. So the second process was, that is the fragmentation of DNA by the restriction enzyme. Fine. Now the third step. Third important process in the recombinant DNA technology, that is separation and isolation of desired DNA fragment. And it is done with the help of agarose gel electrophoresis. You know, DNA molecules, they are negatively charged. So the fragments of DNA molecule, they are forced to move towards the positive charge in the electric field through the matrix, and this matrix, it is made up of agarose. Agarose is obtained from seaweed. How agarose is converted into gel? So for that, agarose is dissolved in the boiling water. When it cools down, it forms double helices. Later forms filament, and these filaments cross-link to form gel. And the pore of the gel, it depends on the concentration of the agarose. DNA fragments, they are separated according to the sizes through the sieving pore, through the sieving pores provided by the gel, and smaller the size, farther it goes. So this is how we separate the different fragments of the DNA. Once the DNA fragments are separated, since it is not visible with the naked eye, it needs to be stained, and for this, that a stain is used, that is called ethidium bromide. So when it is stained with the ethidium bromide and observed under ultraviolet light, then the DNA appears as bright orange bands, and this is how we see the DNA fragments on the gel. Now isolation of desired DNA fragment, and for that the band of DNA is cut out from the agarose gel and it is extracted from there, and that process is called elution. After this, the fragment of desired DNA is purified and used in the construction of recombinant DNA by joining with competent vector. This was about the separation and isolation of desired DNA fragment. In the next video, I will discuss about amplification of gene of interest by PCR, that is polymerase chain reaction.